Good morning, my name is Ana Araujo and my group is confirmed by Sara Gudelo. Today we're going to talk about the development of Etf-3-specific nanobodies to prevent Ehrlichia infection and LNP messenger RNA delivery in cellular and marine models.
So first, what is Ehrlichia cafensis? Ehrlichia cafensis is an obligatory intracellular bacterium that replicates within membrane-bound early endosome-like inclusions in human monocytes and macrophages. Ehrlichia acquires all nutrients from the host, including amino acids, lipids, intermediate metabolites, and iron, for its survival and replication. So, what is Etf-3? Ehrlichia translocator factor 3 binds ferritin light chain, FTL, and induces ferritinophagy, a selective form of autophagy, thereby significantly increase the cellular level iron pool (LEAP) for use by Ehrlichia replication.
What is its mechanism of action? Nanobodies are derived from camelid immunoglobulin G, which lack light chains only consist of heavy chains. This structure grants them a smaller size and greater stability compared to conventional antibodies. Nanobodies act against the Etf-1 factor by inhibiting its function, preventing ferritin autophagy, and thereby altering bacterial pathogenesis. For their administration, the encoding messenger RNA is encapsulated in lipid nanoparticles, allowing the expression in the cell.
So, the general objective is development of Etf-3-specific nanobodies to prevent Ehrlichia infection and LNP mRNA delivery in cellular and marine models.
The study used Western blot, which is a technique used to detect and quantify specific proteins in a biological sample. It's based on protein separation by size using gel electrophoresis, transfer to a membrane, and subsequent detection with specific antibodies. The effects of nanobodies on E. cafensis infection in HEC 293 cells were analyzed. The presence of nanobodies was evaluated to determine whether it reduced the expression of certain bacterial proteins. Quantitative polymerase chain reaction is a technique used to amplify and quantify DNA in real time. Unlike conventional PCR, it allows the measurement of DNA quantified in the sample through the amplification process, providing more precise and quantifiable results. RNA is extracted and converted to cDNA and analyzed by RT-qPCR using SYBR green. The expression of bacterial and cellular genes is quantified to study the impact on nanobodies of infection and ferritinophagy.
So, on the methods, first, 2.4, periodic plasmid construction and purification of recombinant Etf-3. So, the plasmid is used as a vector to introduce and express genes in a bacteria and eukaryotic cell. It's a circular extrachromosomal DNA molecule that replicates independently in the cells. So, once the gene has been inserted into the expression plasmid and transferred into a suitable cell line, the production of the recombinant protein is induced. So, in the study with nanobodies to prevent Ehrlichia infection was mostly used to clone the ETF protein gene from Ehrlichia cafensis and purify the protein for use experiment. Second is 2.7 immunofluorescence labeling. That identifies the presence of protein in a sample by labeling. That's with fluorescent region in the study of Ehrlichia-related ETF protein. Determine the interaction between the use of nanobody with proteins that are related to Ehrlichia infection. So cells were fixed and stained with specific antibodies for Etf-3, ferritin, GFP, and HA. It's a label of the nanobodies.
En el panel A de la figura 5, los investigadores nos muestran un Western blot, el cual de este lado nos muestra una célula que no está infectada con Ehrlichia, mientras que de este lado está infectada con Ehrlichia, específicamente con el factor 3 marcado. Entonces, se analiza el efecto de los nanocuerpos N48 y N51 sobre la degradación de la ferritina, teniendo como en cuenta las cadenas ligeras de ferritina. Esta autofagia es inducida por el factor 3, que en ausencia de los nanocuerpos se reducen los niveles de ferritina, como podemos ver en el control y en el N59. Mientras que en el lado que están los nanocuerpos 48 y 51 se ve que está marcada la cadena. Por ende, podemos decir que la degradación de la ferritina se bloquea, demostrando que estos nanocuerpos inhiben la función del factor 3 y pueden interferir con la proliferación bacteriana. En el panel A de la figura 8, los investigadores nos muestran cómo los nanocuerpos afectan la autofagia inducida por el factor 3 de esta bacteria. Se observa que el lado marcado con el factor 3 incrementa la conversión de LC3 a LC3-2, lo que indica una activación de la autofagia. Sin embargo, en la coexpresión de los nanocuerpos 48 y 51 no parece haber esta conversión. Por ende, podemos decir que los nanocuerpos inhiben la formación de autofagosomas y por lo tanto bloquean la ferritinofagia.
En este caso tenemos el resultado de la inmunofluorescencia, que nos indica la localización intracelular de N48, N51 y N59. que con afinidades relativas para la reunión al Etf-3 se realizó el uso de la línea celular endotelial de mono, que es la RF6A. Se utilizó para el análisis de la localización inequívoca, logrando observar que el Etf-3 con N48, N51 y N59 se localizaba en puntos específicos y podía tener diferentes tamaños. De modo que, aunque los 3 nanocuerpos pudieron localizarse con el Etf-3, solo N48 y N51 lograron tener más afinidad con este en las inmunofluorescencias. Entonces, pues podemos ver que está más presente, que es el compuesto verde fluorescente, con estos dos nanocuerpos, a diferencia del N59. Bueno, la figura 6A es una electroforesis, en la cual, como ya habíamos mencionado antes, la infección por Ehrlichia induce una degradación de ferritina endógena, que promueve el aumento significativo de FTL, Ferritin Light Chain celular, lo que promueve la proliferación intracelular de la Ehrlichia cafensis. Se estudia como N48, N51 y N59, que son los nanocuerpos, afectan la degradación del FTL. Como se observa en esta electroforesis, la proteína de membrana P28 está más presente en el nanocuerpo N59 junto con el control, y esa proteína es de la Ehrlichia, indicando que sigue habiendo infección y no se inhibe esta adentro de la célula. Entonces, N48 y N51 tienen presencia en el FTL, que es el Ferritin Light Chain, lo que significa que pueden degradar al mismo por su afinidad, evitando la proliferación de Ehrlichia cafensis. La actina es una sustancia de control que está en todos los nanocuerpos.
First, we have Lina 2021, who worked with Ehrlichia japonica models for studying A. cafensis infection. We have Mencias at 2014, who described NC0A4 as a receptor that mediates proteinopathy. And for last, Ohashi at 1988 found that Ehrlichia doesn't cause visible symptoms in immunocompetent mice, so all of them agree with the study.
Finally, the conclusions. First one, nanobodies are currently essential for the inhibition of infection caused by bacteria, such as Ehrlichia. Nanobodies which have an affinity or not to proteins that are determined with Etf-3 will determine the inhibition or achievement of the bacterial infection. The second one is the use of Western blurring is more specific for studies in which it is necessary to promote the presence of some type of substance mixture of compounds in a specific site, in this case in action with nanobodies. The action of nanobodies on the specific substance. Third, the molecular biology allows the identification genes essential for the survival of Ehrlichia, which can lead the development of new and more specific antibodies to target therapies against the bacteria, like that.